great pleasure for me to introduce Molly Przeworski. And I tried digging up amusing anecdotes from Molly's past, but unfortunately, Molly has not been forthcoming. However, I do know that she grew up in France, although you couldn't necessarily tell from her accent. She left when she was 18. She went across the pond for the first time in a westward direction, doing an MA. Thank you. Doing her BA in mathematics at Princeton, before going on to do a PhD in evolutionary biology in Chicago, where she worked with Brian Charlesworth and Dick Hudson. Then went eastwards across the pond to do a postdoc in Oxford with Peter Donnelly. Moved to continental Europe, to Leipzig, to the Max Planck Institute of Evolutionary Anthropology for a couple of years, a research scientist, before she started her faculty career back in the US. And she spent most of her early faculty career back in Chicago, in fact. And that's where she became a Howard Hughes early career scientist and was part of what I still think of as a kind of the dream team of population genetics. This great community together with Jonathan Pritchard, Matthew Stevens, Anna D'Arienzo, Carol Ober, Yoav Gilad, others, but a fantastic environment that was incredibly productive and very fun. And then in 2014, she moved to Columbia, and where she's a professor of biological sciences and systems biology. But now she's currently back in France, having gone back over the pond again, spending a year in Paris at the Collège de France. Now, Molly is as you will all know, is a population geneticist with very broad interests. So every facet of population genetics interests her, and she's spent many years working on each of them, particularly recombination, selection, and I think today we'll hear more about mutation. And Molly seems to know exactly the right time to kind of really invest in focusing multiple years and long-term collaborations in investigating one or more of these fundamental processes of population genetics. So that's brought her many awards. She's the Rosalind Franklin Young Investigator Award, the Frederick Willem Bezel Research Award, the Alfred P. Sloan Fellowship, and which I knew when I Googled Molly Przeworski and awards, it was going to be a long kind of task. However, but critically, Molly's kind of caliber of science is very thoughtful and really transcends the theoretical population genetics to analyzing large data sets and making sense of them, not being afraid to ditch the theoretical models and think about the real data and then go back to the theoretical models. And that's been incredibly productive for her. And just three kind of pieces of Molly's work that you know, if I recall off the top of my head that have really kind of had an impact on me is her work on variation in fine-scale recombination patterns, both in humans and in primates. Her work on a really relatively unstudied topic of actually estimating the prevalence of recessive lethals in the population, still something about which we know nowhere near enough. And most recently, really clear thinking about what it means to observe genes in the population that are incredibly intolerant to loss of function variation. A really striking observation, and starting to put it on a much more strong and theoretical backdrop that allows us to think about how we move forward with this interesting observation. So Molly is part of that dream team in Chicago. The nature of her work is that she forms these long-term collaborations. People want to work with Molly, and they want to work with her again and again and again. And so it's no great surprise that her most recent award was actually Outstanding Teaching and Mentorship at Columbia. And Molly is always willing to invest the time, whether it be a grad student needing some help, or whether indeed it is the Sanger Institute needing a site visit committee and someone to sign up for reading thousands of pages of quite tedious documents. So, Molly, we welcome you from your trip to France and your hopefully enjoying the Brexit Schadenfreude. But we welcome you here and hear about your science. Thank you very much, Molly.
So thank you very much, Matt, for the introduction, and I'll know enough not to answer the rhetorical question. about Brexit. So thank you to the other organizers as well for inviting me here today. It's an honor to be here, obviously. As Matt mentioned, I'm a population geneticist. And a distinct feature of population genetics, I think it's fair to say with regard to most other disciplines of science, is that we're in the somewhat unusual situation of knowing what it is that we ultimately need to understand. If we want to make sense of the evolutionary sources of heritable variation, of the differences, say, among the individuals in this room, then we need to understand the processes that gave rise to differences among their ancestors by mutation. We need to understand how these differences were shuffled into new combinations by recombination. And of course, we need to understand how and why they persisted, whether by chance, by genetic drift, or because they were in some way beneficial. And although these processes and their interactions are extremely complicated, and almost in all cases we don't know how to parameterize them, we do know that they comprise the whole picture, which means that we have kind of an end game in mind, and we can work on different aspects of this in isolation, work on meiotic recombination, on natural selection. As Matt mentioned, that's what most of my work has focused on, but with this big picture in mind, which I find quite satisfying. However, today I'm not actually going to talk about this big picture view of population genetics. Instead, I'm going to tell you about comparatively much more recent work that we've been doing in the lab, trying to go back to the kind of ultimate source of heritable differences among us, germline mutations, and particularly human germline mutations. And what I'm going to tell you about today is, unfortunately, both for me and for you, not satisfying, although I'm hoping it'll be interesting. In that, what I'm going to argue is that we know much less about human germline mutation than one might hope and think, and in particular than I thought just a few years ago. And in particular, we don't really know how they arise, with implications that ripple through all kinds of other processes that we might be interested in. And so just a warning up front that this is not at all a typical keynote talk, in the sense that I'm not going to present the arc of a project that's largely completed in which the pieces mostly fit together. Instead, I'm kind of deliberately showing you some recent results in the hope of leaving you as perplexed about germline mutation as I have now become. So to be clear, what I mean by germline mutation are those mutations that accrue between zygote and sperm, in this case, or zygote and egg, i.e. that can be transmitted to subsequent generations. And these mutations can arise in the future parent, either in cell lineages ancestral to the germ cell, or of course in the germline itself. And today I'm going to focus entirely on point substitutions, changes from one letter to another, so I won't talk about CNVs, and I won't even talk about indels. So of course, mutation is a fundamental interest because, as we all know, it is the ultimate source of all heritable diseases, however difficult and tragic their consequences are, and also, remarkably, the ultimate source of all adaptations, however elaborate and intricate they may seem. But understanding understanding mutation is not just of fundamental importance, it's also of kind of practical importance for many fields. So, for example, as you all know, in evolutionary biology, we use the mutation rate as a sort of metronome by which we date when events have happened in the past, say when humans and chimpanzees first split. But to use the mutation rate as a clock, we need to know at what rate it ticks and also how stably it ticks over time. In human genetics, there are a number of contexts in which questions about mutation become important. For example, in evaluating the role that mutation might play in contributing to the increased risk of certain diseases, say schizophrenia, in children of older fathers. Another context in which knowing about the mutation rate is important is in the context of genetic counseling. If a parent comes with a child that has a severe de novo mutation and wants to know what the probability is of a subsequent child inheriting that same mutation, as will become more apparent subsequently, that answer to that question depends fundamentally on what the age and the sex of the parent from which the first mutation was inherited. And so all of these different questions of how mutations accrue over time, with age, with sex, and over development are all inextricably linked, as I will hopefully illustrate to you, and they boil down to this question of how germline mutations arise. So what I'd like to argue today is that despite what we all learn and what many of us, including myself, teach, we don't in fact have a good answer to that question. It's not for lack of studying mutation. So in fact, in addition to countless studies in biochemistry, there have been decades and decades of work on this in both evolutionary biology and human genetics. So for instance, the first estimate of the human germline mutation rate that we have comes from this paper by JBS Haldane that Hillary also presented, in which what Haldane did is he assumed a population genetic model that he and others developed, mutation selection balance, that models the incidence of a Mendelian disease as a balance between the input of disease by mutation and the elimination of disease mutations by selection. And then he focused on very severe diseases where affected individuals don't reproduce. And he inferred what the mutation rate must be to give rise to the incidence of the disease observed in populations. And on that basis, obtained actually almost exactly what we think that mutation rate is many decades and sequencing lanes later, namely around 10 to the minus 8 per base pair per generation. So in addition to the historical importance of this paper, I think it illustrates another distinctive feature of population genetics, which is that it's a subdiscipline of biology in which models matter and thought experiments matter, and in which data analysis is both informed by and in conversation with specific mathematical models. And so just to give you another example of that, the first systematic evidence that we have that most mutations comes from the male rather than the female germline in mammals actually comes from this kind of interplay between theory and data. Specifically, it comes from considering rates of substitutions on different types of chromosomes, autosomes, Y and X, in this case of this paper, X and Y, that are exposed to different extents to male and female germline, and then using theory by Miata and others, estimating how the rates of substitutions that are observed on these different chromosomes must reflect more male mutations than female mutations. And in fact, based on this kind of approaches, it was estimated that in humans, 3/4 of mutations come from males, again, very much what we think it is today. So of course, all of these highly indirect approaches have been upended by what is a much more direct and, in theory, comprehensive way of studying mutation, which is just to sequence parents, sequence the kids, and look for reads carrying mutations in the kids that are not seen in the parents and therefore must have arisen in the last generation. And the first paper to do so, to my knowledge, in humans is this paper by Lee Hood. Since then, dozens of papers have been published using whole exomes or whole genomes, and almost all of them have used blood from parents and kids. So for example, the largest study of its kind until a few months ago was this paper here by Johnson et al., in which they surveyed over 1,500 trios and reported the distribution of de novo mutations that I'm showing here, where each child inherited something between 40 and 120 mutations from their parents, from mother and father. So the fact that there's now so much data and it seems like the approaches are very direct leads one to think that maybe models are now less necessary to interpret the data because you can sort of stare at it and figure out what's going on. But one of the things I'm going to argue in what follows is that, in fact, models, even really simple models that are really just conceptual thought experiments, remain essential to making sense of what's going on. And in particular, I want to get back to this question of how mutations arise. So in principle, we know from a lot of work in biochemistry that mutations can arise from many different sources, and they can be kind of very roughly put into one of two bins. Mutations could arise from replication errors, from the act of copying DNA itself, for example, misincorporation of a DNTP, or they could arise from damage induced by either exogenous or endogenous mutagens that then gets uncorrected by the next round of DNA replication, leading to a mutation. And of course, this dichotomy is an oversimplification because, in particular, these two processes can interact. So the act of copying DNA could itself induce damage. But it's a useful sort of first approximation. So in vivo, in the mammalian germline, which of these forms of mutagenesis predominates? Open almost any textbook, and what you will read is that most mutations in the germline, as opposed to the soma, are due to replication errors, to chance mistakes made in the process of copying DNA. And the reason for this belief kind of stems from two main lines of evidence. The first is actually a really indirect observation from evolutionary biology called the generation time effect. And this is one that was first made in the 1980s, but I'm showing you a somewhat more recent plot here from Melissa Wilson-Sayre's PhD work. So what I'm showing you here is just the phylogeny of a set of mammalian species, and the branch legs here represent the number of substitutions that each species has accrued since the common ancestor. And you can notice that short-lived species, such as rodents, have longer branch lengths, i.e. have experienced more substitutions than shorter-lived species, such as primates. And this observation is not just seen across mammals, but also within mammalian groups. So for example, a couple of years ago now, Priya Moorjani and Eduardo Amorim in my group showed that if you look at different primate genomes and you focus on putatively neutral sites in these genomes, then it looks like old world monkeys have a branch length that's about 40% longer than that of apes. So there's a number of possible explanations for this observation, but the one that was put forth in the 80s and stuck was that it has to do with the number of cell divisions that these species go through per unit time. And the reasoning is that if all these different species have undergone similar numbers of cell division between zygote and germ cell, then being more short-lived necessarily implies that you go through more cell divisions per unit time. And if mutations are due to errors in replication, then going through more cell divisions per unit time means accumulating more mutations per unit time and hence more neutral fixations. So this highly indirect observation that mutations accumulate more rapidly in shorter-lived species was taken to support the notion that mutations are due to errors in replication. But of course, the much stronger line of evidence that leads us all to think that the mammalian germline mutations are mainly replicative in origin comes not from a comparison of different species, but from a comparison of human males and human females and the very divergent developmental trajectories that their germlines take, right? So in mammals in general, in humans in particular, oocytogenesis is completed by the birth of the future mother, and it's estimated that there are approximately 15 cell divisions between sexual differentiation and the formation of primary oocytes that are then in stasis until ovulation and fertilization. In contrast, in males, it's estimated that there's maybe 20 cell divisions or so until spermatogonial stem cells, and with very few, if any, cell divisions before puberty. And then, of course, after puberty, spermatogonial stem cells are continuously dividing at an estimated rate of 23 divisions per year. Now, these numbers are pretty fuzzy because they're based on somewhat indirect estimates that make strong assumptions. But nonetheless, what's clear is that there's this marked difference in the number of cell divisions that the two germ cells go through. So in particular, if you think about the number of cell divisions that germ cells of future parents have gone through as a function of their age, then if parents could reproduce at birth, which in humans obviously they cannot, they would have gone through very similar numbers of cell divisions. If you think of mothers and fathers reproducing shortly after puberty, they too would have gone through very similar numbers of cell divisions. But by the time the parents are 30, the male germ cells will have gone through an order of magnitude more cell divisions than the maternal one, suggesting that if mutations are replicative in origin, then their accrual should look something like this picture. Not necessarily exactly like this picture, because per cell division mutation rates may vary over ontogenesis, but something like this picture. So in particular, most mutations would come from males, and there should be a linear increase in the number of mutations with paternal age. And that's indeed what we see. So this is a figure from a paper by Augie Kang and colleagues published in 2012. And in these data, 3/4 of mutations are paternal in origin. And you can see that there is a linear increase in the number of mutations with paternal age, where the paternal age at conception of the child is on the x-axis, and the number of mutations inherited from both mother and father on the y-axis. So in fact, the existence of a paternal age effect was already hypothesized by Weinberg close to a century ago now, and long before we had next generation sequencing data, seemed to lend strong support to this notion that mutations are replicative in origin. In particular, Jim Crow, in his authoritative review on the topic, explained that the male mutation rate in human males, especially older males, is generally much higher than in females, mainly because in males there are many more germ cell divisions. So that second line of evidence that mutations accumulate with paternal and not maternal age seem to sort of clinch the case for mutations being due to errors in replication. Of course, although meanwhile, people appreciated that it's not the case that all mutations are due to errors in replication. In particular, we know that the deamination of CpGs, a topic that actually a number of talks have covered in previous days, is a source of mutations that are non-replicative in origin. So just to remind you, if the CpG is not methylated, then when it spontaneously deaminates, it deaminates to a U. And that is actually, there's actually a glycosylase that's specifically dedicated to the recognition of that U, so it's quite efficiently recognized and repaired. However, when the methylated CpG deaminates, it deaminates to a T, which is a natural base and is not as efficiently removed, more frequently leading to mutations. So what do we expect in this case? In the case where the mutation arises from damage that is not repaired by the next round of DNA replication, rather than by replication error itself, how do we expect those mutations to accrue with age and with sex? When my former student Ziwei Gao and I started to think about it, we realized we didn't know. And so she wrote down a simple model that it's in its kind of vanilla form best applies to these methylated CpGs. She considered that there might be a fixed instantaneous error rate, mu, at which methylated CpGs deaminate, therefore, to a T on one of the strands, and then if unrepaired by the next round of DNA replication, one of the daughter cells will inherit that mutation. She further considered a fixed instantaneous repair rate, R, and in the particular version of the model that I'm showing you, that when repair occurs, it always occurs correctly, meaning that if repair occurs before the next round of DNA replication, no mutation ensues. So the full model looks something like this, and she can solve it for the proportion of mutated bases as a function of time since the last cell division. And what's interesting is that looking at this model, she realized that there's kind of two limits. One limit in which repair is highly inefficient relative to the length of the cell cycle. And in that limit, as is intuitive, to a first-order approximation, the repair rate doesn't matter, and therefore the proportion of mutated bases accrues proportionally to absolute time, which means that mutations actually track absolute time in this assumption of a fixed damage rate. The other limit, however, in which repair is really efficient relative to the length of the cell cycle, actually leads to an asymptote where the proportion of mutated bases no longer depends on the time since the last cell division. And what's interesting about this other limit is that in that case, even though the mutations are non-replicative in origin, they actually still track cell divisions. And the intuition here is that the few errors that tend to become mutations tend to be those that occurred right before the DNA replication. So what's interesting about this model, obviously, in all likelihood, we lie in neither of these two limits, and in addition to which the model is obviously a gross oversimplification. But I think the model does illustrate two things. So the first thing that it illustrates is that even non-replicative mutations can, under certain conditions, track cell divisions. The second thing that it underscores is that the repair efficiency is a key parameter in determining what we should expect, how we should expect mutations to accrue with age. So this first conclusion that non-replicative mutations can track cell divisions when highly efficiently repaired is relevant to the interpretation of germline mutations, but also somatic mutations. So for example, I'll draw your attention to this paper published in 2015 by Tomasetti and Vogelstein that many of you may be familiar with, in which they reported, and this is a figure from their paper, that the lifetime risk of various incidents of cancer is a function of the total number of stem cell divisions. And they interpreted this pattern as saying that basically there are more mutations when there are more cell divisions, more mutations that lead to cancer, and therefore they concluded that these random mutations arising during DNA replication in normal non-cancerous stem cells drive the majority of cancer incidence. A conclusion that was baffling to the field for a number of reasons, but in particular because we know, of course, that exogenous mutagens have a big effect on the incidence of cancers, for example, tobacco, UV, and so forth. And what our model, I think, clarifies is that even if you were to grant them this picture, even if you were to grant them interpreting the picture in terms of more driver mutations arising when there are more stem cell divisions, it would be incorrect to infer from that that mutations are replicative in origin, because under some conditions, even non-replicative mutations, such as those that are due to UV damage or smoking and so forth, can track cell divisions if very efficiently repaired. in the context of germline mutations, the model helps to specify what we might expect to see in terms of the patterns of the number of mutations as a function of maternal and paternal age. So for example, that if mutations are replication driven or repair is very efficient, we would expect them to accrue with paternal but not maternal age, whereas if mutations are due to inefficiently repaired damage, we might expect them to accrue with just age, with absolute time. Now, that doesn't mean that they're going to accrue at the same rate in the two sexes, because the damage rates might in fact be quite different in spermatogenesis and in oocytes. And here I've hypothetically drawn the damage rates higher in males. So what this model makes clear, I think, is that you cannot infer from the fact that there's a paternal age effect what the source of mutations is, because it could in fact arise from any of those scenarios, a point that was already made by Muller in the 1950s but kind of lost in the literature. However, if you see a maternal age effect, that is evidence about the source of mutations, because mothers do not have any cell divisions after birth. And so it was therefore big news when Johnson et al. Reported not only a paternal age effect on mutations, but a maternal age effect. So what I'm showing you here is the central figure from their paper, in which now each child is represented by 2 dots, paternal mutations in blue, maternal mutations in red. And you can see that they again see a linear increase in the number of paternal mutations with paternal age, with an extra 1.5 mutations per year of age of the father. But they also see a significant increase in maternal mutations with maternal age, and a non-negligible one, since it's about a third the rate. So they interpreted this finding as indicative of the fact that the sex difference you see here reflects the relative lack of mitosis in aging oocytes compared with spermatogonia, which may enrich for damage-induced de novo mutations. In other words, what they're saying, I think, is that in oocytes, what you're seeing is the result of damage, but in males, what you're seeing is the result of spermatogenesis and cell divisions leading to mutations. What's a bit puzzling about this interpretation is that if you look at the mutation spectra, the types of mutations inherited by the two sexes, although there are differences, they're very subtle differences. And it's kind of hard to reconcile that with these mutations having drastically different sources in the two sexes. And so on the basis of this observation, we decided to test this hypothesis that it's really spermatogenesis that drives the male bias in mutation in humans, just by reanalyzing their data. And specifically, we decided that we would consider parents of older and older ages and see whether the male bias is really increasing. So that's what I'm showing you here. And to our surprise, we see absolutely no increase in the rate. So what I'm showing you on the y-axis is just some raw data, the fraction of paternal mutations among all phase mutations for parents of roughly the same age. And you can see there's no increase in the degree of male bias with parental ages. And in fact, it's already 3-to-1 by puberty, more or less. So first I should say that we found this in the decode data, but just to make sure that it wasn't a quirk of their data, we reproduced it in the data published by Wang et al. And 2 other data sets. So we're seeing it in every large data set, every data set that's large enough to be powered to detect a maternal age effect. So this is not an optimal use of the data. We can also kind of take a model-based approach and do a little better, and that's what we did. So we basically estimated sex-specific rates of increase in the two sex-specific rates of increase, using maximum likelihood, basically just reproducing what Johnson et al. Had done originally, using both phased and unphased mutations. And when we do that, we can estimate the ratio of male to female mutations as a function of parental ages. This is again showing it to you for parents who are roughly the same age, and the gray denotes our uncertainty. And so you can see that even using all of the data where we have a large amount of power to detect any increase, we don't see an increase in the male bias with age. So what's going on here? So there are several possible explanations. One explanation is that there are actually more cell divisions in male by puberty than has been estimated. In other words, that male germ cells have already undergone more cell divisions by puberty than female germ cells, which is why the ratio is already 3-to-1. Another possibility, not mutually exclusive, is not that there are more cell divisions, but that each cell division in males is somehow more mutagenic, and so you end up with more male mutations than female mutations by puberty. Both of those explanations could account for why the ratio is 3-to-1 by puberty, but doesn't readily account for the stability afterwards. In fact, these explanations to account for the stability afterwards require a bit of a miracle where a whole bunch of per cell division mutation rates and cell division rates mysteriously balance out. So it's mathematically possible, but it requires a huge amount of special pleating. The other possibility, which seems more parsimonious to us, is to hypothesize that in males too, most mutations are actually due to damage, which would also help to reconcile this observation that their mutation spectrum looks pretty similar. So to investigate this in a little bit more depth, we decided that we would look into two cases where there was some independent information of a non-replicative source for these mutations and see whether what we're finding was consistent with this independent evidence. So the first case we considered are methylated CpGs. And what's interesting about methylated CpGs is that they too have very different developmental trajectories in males and females, because after methylation marks are deliberately erased, after sexual differentiation, the male germline is almost immediately remethylated. In contrast, the female germline is actually not remethylated until shortly before ovulation, which means that if you consider older parents, say parents who are 40, then in the male, the germline has been almost continuously methylated with plenty of opportunities for spontaneous deamination, whereas that we considered are C to Gs. Much less is known about this, but Johnson et al. Had this really interesting report of a number of observations I don't have time to go into that together suggested that they might be associated with the repair of accidental double-strand breaks and enriched in aging oocytes. And so consistent with their hypothesis, when we reanalyzed the data, we found that C to G mutations tended to be found on the same chromosome as a deletion of more than 5 base pairs more often than expected by chance, and we're actually closer to these de novo deletions than other types of mutations. And in work that my student Ipsita Agarwal presented to you last year, we also found some evidence that the same mutational signature was associated with deliberate double-strand breaks that are made very late in meiosis. And so potentially indicative of the fact that these C to G mutations are associated with both spontaneous double-strand breaks and a subset of deliberate double-strand breaks. And so in this case, we actually see an exponential rate of increase of these C to G mutations with maternal age, reflecting an accelerating rate at which they seem to be accruing in oocytes and a decrease in the male bias with age. So those are two mutation types where we have some independent evidence that they're not replicative in origin, and that together already account for 1/4 of mutations. What about the other 3/4? Do we then see that they start to track cell divisions? So this is what we see excluding these two types, and you can see, if anything, there's even less of a relationship to paternal age than in these two other cases. So these mutations don't seem to track cell divisions either. So it seems to us that because mutations accrue with both paternal and maternal ages, and it turns out that the ratio of those slopes is pretty much fixed, and therefore there's no increase in the male bias with parental ages, spermatogenesis cannot be driving the male bias in mutation. What about the other line of evidence that had been marshaled in support for mutations being replicative in origin? Recall that different species evolve at different rates. In particular, apes seem to evolve more slowly than old world monkeys, such as baboons. In particular, human females have a generation time that's maybe 28 years, whereas baboon females, it's more like 10. And of course, baboon males reach puberty and reproduction earlier, but I thought to have a higher rate of spermatogonial stem cell divisions, maybe 180 cell divisions between puberty and reproduction on average, versus about twice that number for male life history traits. So if it's the case that mutations track cell divisions, then if we think again that there's the same number of cell divisions between between zygote and egg, then we might expect the mutation rate per generation to be similar in human and baboon females. And we might also expect that the ratio of male to female mutations should be higher in humans than it is in baboons, because males have undergone 2 times more cell divisions. And so we decided to test that hypothesis by this kind of direct approach of resequencing pedigrees from baboon and from humans. And we did that in the 2 species so that we could make a direct comparison using the exact same pipeline and focusing on orthologous regions of the genome in these 2 species. And so when we do this in humans, we obtain for the parental ages in our sample a sexed average mutation rate of 1.22 times 10 to the minus 8, which is actually very similar to what you would get from the Johnson et al. Study if you plugged in the same ages. However, interestingly, what we get in baboon is actually 2.5-fold lower. Namely, for the parental ages in our sample, we instead get about 0.5 times 10 to the minus 8. So what of our predictions that if mutations track cell divisions, mutation rates per generation should be similar in human and baboon females? They're not. In fact, they're about 2.5-fold higher in human females than baboon females. What of our prediction that if mutations track cell divisions, the proportion of male mutations should be higher in humans? It's not. In fact, it's very similar in the two species. So neither of those are consistent with at least our naive expectation if mutations are tracking cell divisions. However, they are readily explained if mutations are damage-induced, at least predominantly damage-induced, and damage rates are similar in the two species. Namely, our female mutation rates basically reflect the average ages of reproduction, a 2.5-fold difference instead of 2.8, and we would expect the male bias in mutation to be roughly the same in baboons and humans. More generally, if we zoom out to all the mammals for which we have direct estimates of the ratio of male to female mutations, then the point estimates are all remarkably similar, despite the fact that these species reproduce after months, years, or decades. So again, not consistent with the notion that the degree of male bias is tracking the number of mutations in males instead of females. So putting this together, what have we learned? I think first that it's not always the case that only replication-driven mutations will track cell divisions. Under some conditions, damage-induced mutations can track cell divisions as well. Second, it looks like the male bias in mutations in humans is already present by puberty, so spermatogenesis is not driving the male bias in mutations. And at least in a comparison of human and baboon, it looks like maternal mutation rates reflect ages, potentially consistent with them tracking absolute time as well instead of the number of cell divisions. And moreover, based on at least 5 mammals, the fraction of paternal mutations appears to be roughly the same. So together, I think these lines of evidence suggest that actually mutations aren't tracking cell divisions after all. And they raise the possibility that, in fact, germline mutations, too, are predominantly due to damage. And that immediately raises the question of what kind of damage, and in particular, whether it's mostly endogenous or exogenous. For example, could it be that individuals living in very different environments actually experience different mutation rates, as proposed in a preprint, for example, that reported that the Amish have lower mutation rates? What I think this summary also highlights is where the real gap in our understanding lies, which is in development. Because we should expect these kind of balance of replication, damage, and repair to vary over ontogenesis as it does for CpG transitions and C to Gs. And so we want to know how these different mutation types accumulate over development in order to answer these different questions. And in that regard, I just want to kind of bring up a detail that I've swept under the rug until now, which is that when we sequence blood from parents and children, we don't actually study mutations that have accrued between zygote and germ cell. We study mutations that have occurred after imposing a bunch of bioinformatic filters that we have to impose to get rid of false positives. We study mutations that have accrued in the germline of the parents and in the early development of the child. And it's interesting that a number of recent papers, these and some others, have actually highlighted that at least a few % of the mutations that we're detecting in these trio studies are arising from the early development of the child. Now, how is that confusion possible, you may wonder? It's possible because what we call a paternal mutation is actually just a mutation on the paternal germline, on the paternal genome, sorry. So one that could have arisen in the early development of the child. And so in that regard, I just want to remind you of a couple really interesting features of these first few cell divisions and why we might think that they might be particularly mutagenic. So one of these features is that the two genomes, the paternal and the maternal genome, are actually replicated separately in their own pronuclei in the first cell. And the second, of course, is that in the first couple of cell divisions, repair is completely reliant on a store of RNA and proteins from the oocyte because there's no transcription yet. And so that raises the intriguing possibility that some of the age effects that we've been interpreting as prezygotic are actually postzygotic. And in particular, it raises the question of whether a mother's age, and therefore her oocyte's age, could actually affect how cleanly these first few cell divisions go. And we have some evidence to that effect in this preprint, this paper, posted here. Namely, we think we see an effect of maternal age on the number of mutations that are accruing on the paternal genome in the first few cell divisions. But because that finding is very tentative, I'm not even showing it to you, and I'm preferring to phrase it as a question in the hope that someone will either replicate it or disprove us. The last question that, of course, we want to know is what is going on with the phylogenetic rates? Why is it that we see this variation across species, if not a function of cell divisions? Could it be a function, for example, of damage rates that are varying with life history traits? And so I want to leave you with one more puzzle, which is that when we estimate yearly mutation rates in baboons and humans, we actually get almost identical mutation rates, despite the fact that the phylogenetic rate is 40% longer in baboons than it is in humans. When Matt Hurls estimates the mutation rate in mice compared to humans, the rate in mice is 10 times higher, even though the phylogenetic rate is only 2 to 3 times higher. So there seems to be this fundamental disconnect, not just in humans, which a lot of ink has been spilled on, but in almost any mammal that we have looked at, between the rates estimated at present and the phylogenetic rates, which is all the more puzzling as the phylogenetic rates are actually kind of well behaved within a clade and pretty consistent one with another. So as I've hopefully convinced you, there's a lot we still don't understand about how mutation rates accrue with time, with age, with sex, and overdevelopment. And these seemingly kind of very disparate questions, ranging from really basic science questions about the evolutionary clock or human developmental biology, to these very applied questions of how we calculate recurrence risk for deleterious de novo mutations, are in fact all intricately intertwined and related to this question of how germline mutations arise. And I think the flip side of all of these questions being so intricately intertwined is an opportunity, because I think there's some benefit to be gained by integrating these evolutionary, developmental, and biochemical perspectives. At the end of the day, these different questions, these different pieces of the puzzle have to fit together, so the fact that they don't fit together is telling us that something pretty fundamental is still missing from our understanding. So on that note, I just want to take 2 minutes and thank a bunch of people who have puzzled through these questions with me. I want to single out Laure Segurel and Min Wyman, who indulged the fairly absurd idea I had about learning about germline mutation by writing a very, very long review about it. I want to thank Priya Morjani, who led much of the painstaking data analysis, Felix Wu, a current graduate student in my lab, who extracted signal from the baboon data, and most of all, Ziwei Gao, who's been kind of a sounding board for me all throughout and led a lot of the work that I talked about. And I also want to take the opportunity to thank all of the members of my lab, past and present. I think it's fair to say that at least under the right conditions, being a scientist is an incredible job. And one of the reasons it's an incredible job is because you get to work with these talented people who come from all these different walks of life and all these different horizons, and I've been very lucky in that respect. I've also been lucky in a number of other respects, so I'll ask for 30 more seconds of your time to mention a few of those. So I was introduced to population genetics by some extraordinary scientists, and then I was helped to survive that introduction by fellow graduate students and postdocs with very high standards. And then I was lucky enough to end up at the University of Chicago and more recently at Columbia, surrounded by some colleagues who are both stellar scientists and outstanding human beings. And then all throughout, people who were neither my mentors nor my colleagues sort of randomly were very supportive of me at key moments that made a big difference to me. So these are some of these people with my thanks.
That was great, mind-blowing. So it's known that, at least in some men, sperm accumulate oxidative damage, so double-stranded DNA breaks, and like 8-oxo-DG, for example, and the sperm are missing certain DNA repair machinery, and so could that be an explanation for this? Have you thought about that?
Yeah, so that's a really I didn't plant the question, to be clear. In fact, I find it really intriguing. So if, for those of you who don't know this, fun and bizarre fact that both in mouse and in sperm, both in mouse and in humans, interesting, the last few cell divisions of spermatogenesis, there's no base excision repair, which seems like a poor evolutionary choice. And so the damage that accrues in the sperm then has to get repaired in the zygote. And in particular, there's a lot of oxidative damage, and that can lead to C to A's. And so, in fact, the line of evidence that I most believe for our maternal age effect on the paternal genome is that we see a maternal age effect on the paternal C to A specifically.
Thank you, Molly. If the rules have changed in the last, say, 15 generations or post-industrial revolution, and would this be immediately detectable? Would this be seen in the data in these kind of analyses, or is it possible that that's the shadow in the data sets between the phylogenetic analyses and the more recent mutation detection?
Yeah, so in humans, there's been a huge amount of attention paid to that since a paper that Richard and Alwyn Scally wrote in 2012 to this kind of disconnect between, and a lot of people have looked into that, including my lab, and one of the ways in which we've chosen to do that is by looking at the mutation spectrum estimated, for example, in Johnson et al., and then in singletons, doubletons, tripleton, and so forth, which allows you to kind of travel back in time. And we see some subtle differences, but nothing that would be sort of consistent with a massive shift in what's going on.
That was a lovely talk. I was wondering whether you had used both transcription-mediated repair and also, you know, these transitions between early replicating to late replicating areas where you sort of know which way the replication fork is going. Now, there may be some epic assumptions that, you know, you can take cell line replication data and imagine that is the way spermatogenesis works. But I was just curious because both of those give you some strand bias on the on either replication or repair
Right.
components to this.
Yeah, so not in the context of this project, but in the context of other projects, we've done some work looking at a transcription-coupled repair and exactly the kind of asymmetry in template and non-template strand. In particular, we had a paper comparing what that looks like for germline mutations compared to somatic mutations to see if we could see a difference there. So we've toyed with these ideas. The problem, in my mind, with, for example, the replication timing. So replication timing is a massive effect, as you know, on the massive, no, but it explains a large proportion of the variance of the mutation rate across the genome in large regions. However, of course, their damage and replication interact in complicated ways. So seeing that pattern at least doesn't allow me to know how to disentangle the different sources of mutations.
Hi, I'm wondering whether the mutations from father tend to accumulate in particular genomic region, or are there observable difference between, say, the mutations in semen with compared with those in actual offsprings?
Right, so for example, could there be some sperm selecting acting on the mutations that arise? I think that's a super interesting question. I don't have a good answer for you in the sense that we've done some back of the envelope calculations that lead us to think that it doesn't seem plausible that that would be a massive effect, but it's certainly something that is worth thinking about. We don't see, for example, a deficiency in exons or in functional regions compared to what you see, what you might expect to see. It looks like base composition plus some other elements like replication timing and so forth explains things pretty well, but I think that's definitely an area worth looking at in more depth.
Last question up there.
A follow-up to the first question. So given the intriguing idea that you may have a burst of attempted repair of unrepaired lesions after fertilization, are there differences in mutation rates as calculated by looking at single-cell sperm sequencing versus looking at trios of liveborns?
Yeah, so to my knowledge, and someone in the audience is free to correct me, the only sperm-based estimate I know of from DNA is this older paper by Steve Quake from 2012, I believe. And you know, that was a very limited sample that got an estimate that is something like 1.2 times 10 to minus 8 for age 40 or something like that. So, I mean, not actually, the number I cited is wrong, so ignore that. But they got what people are supposed to get for an age of 40. And I don't know of other single cell DNA sequencing based estimates. But again, I may have just missed something.
Great. Well, we'll leave it there. Many more questions to come over coming years. And just thank Molly again.